Hello, everyone. Thank you for coming today. I'm excited to share some of my research with you. When you find a plant in nature, the first things you probably notice are the shape and the size, the colors of that plant. If it has a flower, maybe you notice if that flower has a nice scent. But some of the things you probably don't notice are all the different organisms that associate with that plant. Some may have negative effects. There may be insect herbivores that eat the plant tissues. There may be pathogens that attack the plant. But a lot of them are likely to be beneficial. And these include the mycorrhizal fungi that associate with the roots of the plant and help it to gain essential nutrients for its growth. It could also be endophytes or phylosphere organisms, the bacteria and the fungi that live on and inside of the plant tissues and can help protect it from herbivores or from pathogens or from other stresses. And so what I study are all these different associations, the community of organisms that live within an ecosystem. And I'm going to be talking to you today about carnivorous pitcher plants. I'm showing you one here. And this pitcher plant associates with maybe even more organisms than your average plant, because inside of it, it hosts this little aquatic ecosystem. So a pitcher plant is actually a modified leaf that over evolutionary time has formed a cup. And that cup holds a mix of rainwater and plant secretions. And the plant produces a nectar around the edge of this pitcher. And that attracts insects, primarily ants, which feed on the nectar and then they fall in and drown in the pool inside. It's very slippery and they can't climb out. And once they drown, they're broken down and decomposed. And the plant is able to use those insects to get important nutrients like nitrogen and phosphorus that are not in high abundance in the areas that it grows. But in this little ecosystem, even though this plant attracts and eats insects, it also hosts other insects that are only found in pitcher plant microcosms, and a whole community of bacteria and rotifers, fungi, protozoa. All these different organisms live in this little microcosm inside of the pitcher. So today I'm going to first talk about a concept called convergent interactions, and then thinking about how this might be playing a role in a comparison between pitcher plant ecosystems. I'll finally talk a little bit about continuing work. And the majority of my talk today will be in section 2, in this middle part. So starting with convergent interactions. In The Origin of Species, Darwin talked about something that he called analogical resemblances. And we now know that this is what we call today convergent evolution. Convergent evolution is where you see the same form and function evolving independently in different lineages. And an example that he gave in The Origin of Species was the dugong and the beluga whale. These two have a very similar body shape. They have a very similar lifestyle. But Darwin hypothesized that they came from different lineages, and we now know that this is true. So my colleagues and I developed this concept called convergent interactions. And we wanted to do this to think about convergent evolution, not just in single individuals, single species, but among different species in multi-species interactions. So we define this as the independent emergence of multi-species interactions with similar physiological or ecological functions. I'll give you a couple examples of this in plants. So the first example is ectomycorrhizal associations that form between plants and fungi. And in this association, you have a Harding net that forms around the tip of a root. And the fungi are able to scavenge in the soil for nitrogen that would be very difficult for the plant to access on its own. And they exchange that to the plant for carbon that the plant has photosynthesized using its leaves. And this association has evolved independently more than 14 different times. And so it seems like it's a pretty good solution, both for plants and for fungi, for getting essential nutrients that they need to grow. Another example, which you may have heard a little bit about yesterday in Dr. Nomi Pierce's talk, are ant-plant associations. And in this interaction, you have a plant that produces some sort of structure. Here I'm showing hollow stems, but they could also be domatia, swollen thorns, where the ants live. And the ants then have housing and also some food that's provided for by the plant. And in exchange, the ants protect the plants from herbivores, often from other insects that are trying to eat the plant leaves, but it could also be big herbivores like giraffes or elephants. And here I'm showing you two examples, one that is in South America and one that is in Southeast Asia. But they look very similar. So we argue that convergent interactions is a useful concept because it can help us to recognize the ecological role of a symbiosis involving undescribed species. So for example, with ectomycorrhizae, a lot of fungi are still undescribed. But if you find a fungus associated with this hearted net and a root tip, you already have an idea about what that organism might be doing within that ecosystem. And you can predict a function of that species. You can also infer selective pressures acting in one system based on data from another system. So for example, if we know a lot about the ant-plant interaction in South America, and we know that the selective pressures driving that association for the ants are a need for housing, and for the plants are a need for protection from herbivores, then we may be able to infer that those similar selective pressures are probably also acting to form that same interaction in Southeast Asia. We wanted to understand if convergent interactions were happening in pitcher plant communities. Here, what I'm showing you is an evolutionary tree of plants. And what you can see is mapped onto this are all the separate evolution events of carnivory. So carnivory has evolved multiple times across plants. And carnivory generally evolves when plants need essential nutrients like nitrogen and phosphorus, and they're growing in habitats where that is not very abundant. So in order to be competitive in their habitat, they find another way of getting those nutrients, getting them from trapping and digesting prey, mainly insects. And what I'm going to highlight on here are three independent evolutionary events of true pitcher plants. So true pitcher plants are formed by a single leaf, single modified leaf. And there are three different groups of these. There's the Nepenthaceae family that's found in Southeast Asia. There's the Cephalotaceae family. There's one species in it, Cephalotus follicularis, and it's found in Western Australia. And then the Sarraceniaceae family. And there's three different genera, and these are found across the Americas. So you can see that these different families are in different orders of plants, and they're really distantly related. So they're a really elegant example of convergent evolution. And just to highlight this, I'm going to be focusing mostly today on the Nepenthes and the Sarracenia. And the Nepenthes and Sarracenia are really distantly related. So Nepenthes is actually more closely related to cacti than to Sarracenia. Sarracenia is more closely related to blueberries than it is to Nepenthes. So they don't have a common ancestor that has any of these carnivorous traits or even similar shape and form. When we're comparing these independently evolved pitchers, the Sarracenia and the Nepenthes, we were wondering if these convergent hosts lead to convergent interactions with their associated communities. So we know that there's many different organisms living in these pitchers, but a lot of them were not very well known, particularly the fungi and the bacteria that make up their microbiome. So to understand who was there, we did some sampling and some sequencing. We collected over 700 samples in North America, Southeast Asia, and Australia from pitchers and from the bog water or the soil that was directly surrounding those pitchers. We did metabarcoding of the bacteria and the eukaryotes using the 16S and the 18S ribosomal RNA gene sections. And this is a way of understanding who is there. We also did shotgun metagenomes to look at the functional potential of the whole community. So we did this just from a subset of these samples. And this is a way to look at the genomes of all the different organisms present to see what they can do. Here I am collecting in the Gulf Coast of the US and in Southeast Asia on the island of Borneo. And once we had our data, we were wondering if similar organisms colonize both the Nepenthes and the Sarracenia pitchers, even though they're found on opposite sides of the world. We found that there are similar organisms colonizing them. So here what I'm showing you is an evolutionary tree of all the different bacteria, all the different bacterial species from our study. And in brown, around in the outside, these are the different bacteria that were found in the bog water or the soil surrounding the pitchers. And then here in red and in blue are all the different bacterial species that were found in at least 10% of our Nepenthes or our Sarracenia pitchers. So these are the organisms that show up reliably and repeatedly in these pitchers. And you see that they generally do come from the same bacterial families or related groups. You see that they come from these same phylogenetic clades. And you also see that for the eukaryotes. It's a little more restricted here to the rotifers, the dipteran insects. These are mainly specialized mosquitoes and midges that are found in pitcher plant habitats, and the aquatic histiomatid mites. So this was really exciting because it suggests that these related organisms colonizing pitchers on opposite sides of the world may not be the same species, but they're closely related and they may have a lot of the same functional capabilities. They may be fulfilling similar ecological roles in these different little pitcher ecosystems, even though they're on opposite sides of the world. We wanted to know about function. And so looking at these metagenomes that we had, we hypothesized that pitcher plants would be enriched in genes for prey degradation. And in particular, I'm going to show you our data for chitinases, which are the enzymes that break down chitin, which is the main component of the insect exoskeleton. We hypothesized that chitinases would be enriched in pitcher plants as compared with other published metagenomes from other habitats. So from phylosphere, which was leaf surface, lakes, or soils. And we did find that pitcher plants were enriched in chitinase genes. On the other hand, we hypothesized that pitcher plants would have fewer cellulase genes that break down cellulose, which is a main component of plant tissues. And we found that it was true that the communities living in pitcher plants generally had fewer cellulases than found in these other habitats. So they seem to be similar types of organisms that colonize pitchers in Southeast Asia and in North America, and they seem to have similar capabilities. But what about if you put both of those hosts in the same environment? If you had Sarracenia and Nepenthes pitchers growing together in the same environment, would they be colonized by very similar communities? Would they create a similar enough habitat that they would select for particular types of communities? For this experiment, we were using Sarracenia purpurea in the bog where they were growing naturally in Massachusetts. We also had them in pots as a pot control. And then we had multiple different Nepenthes species in pots that were put out in the bog over the summertime with these Sarracenia. We also had sterile glass tubes with or without prey to be able to compare with these pitcher plants. And this is what the setup looked like. So you can see here is a Nepenthes pitcher already catching some prey, some insect prey, a native Sarracenia pitcher that was local to this habitat, and then the glass tube. So what we found from this experiment were that the bacterial and the eukaryotic communities in Nepenthes pitchers were very similar to those in the Sarracenia pitchers, as long as the pH or the acidity of the fluid was similar. So the pH is important because Nepenthes can actually actively acidify their pitchers and make them super acidic down to a pH of 2 or even lower. And when they do this, they really select for only very particular organisms that can thrive in that very low pH. But a lot of Nepenthes pitchers are also much higher pH, around 5, like Sarracenia. And when they had a similar pH of their fluid, they selected for very similar communities. In particular, we noticed that the pitcher plant mosquito, Wyomia smithii, which is normally only found in Sarracenia purpurea pitchers, was colonizing our Nepenthes pitchers in this experiment, and they were able to complete their life cycle. So the Nepenthes pitchers were acting as effective alternate hosts for these communities, which just shows the extent of the convergent evolution of these plants and how that can shape associated communities. So from these experiments, we saw that related species colonize pitchers from Nepenthes and Sarracenia plants on opposite sides of the world. The communities are enriched in degradation enzymes, and they likely have similar functional roles in these little ecosystems in the pitchers. When Nepenthes and Sarracenia plants grow together in the same habitat, their pitchers are colonized by very similar communities. And this concept of convergent interactions can help us to better understand complex species interactions and look at it through a functional lens. Think about what are the important roles these species carry out in their ecosystems, and how might that be affected by this interaction and by evolution over a long period of time. I'm going to touch briefly on some continuing work by highlighting some recent projects in the pitcher plant system. This study was looking at effects of history on bacterial community assembly and function. And I took pitcher plant communities from their pitchers in the wild and brought them into the lab and looked at how their history affected the structure of the communities and the functions, and found that there were some key functions that were really affected by the history of where these bacterial communities came from, where they originated. We also continued these same communities over more than a year in the lab and looked at community dynamics over evolutionary timescales, thinking about how evolution might be acting within these complex communities. One interesting finding we found from this paper was that if you looked just at the species level for some organisms, you wouldn't see any correlation over time. They didn't seem to be an important interaction happening between these two species. But when you have shotgun metagenomes and you have really detailed pictures of the strains that make up these species, you actually see that the minor strains were strongly negatively correlated. And it seemed like there was an important interaction happening between these strains that may affect evolution within these communities. I also just recently submitted a perspective paper with colleagues looking at how a functional trait-based approach can help us understand microbiomes across space and time. And we used pitcher plant examples for this perspective. Some ongoing projects include that I am writing up a paper about looking at the communities of Cephalotus follicularis pitchers. If you remember from the beginning of the talk, this is the species that's the only one in its family, and it's found in Western Australia. And there's not a lot known about these communities. So I'm excited to be analyzing this data. I'm also looking at if bacterial nitrogen fixation in pitchers could be another source of important nitrogen for the plants in collaboration with the Anne lab at University of Wisconsin-Madison. And I'm looking at how different bacterial communities could impact pitcher plant nitrogen uptake from their prey. So if some communities have more digestive enzymes, does that help the plant in accessing nitrogen from its insect food source? This is part of a recent NSF award. So with that, I'm going to come back to this image from the beginning, because I hope that now when you see these beautiful plants in nature and you notice the way that they look, perhaps if they have flowers, the way that they smell, I hope that you also think about all these different organisms that may be associated with these plants and the different ways that they could harm them or that they could be essential for the health and fitness of their hosts. I want to thank so many different people who are involved in this work, the 3 different labs where this work was carried out, also my current lab at Boise State, our international collaborators, the many places that allowed me to do fieldwork and sampling, and the important funding sources that made this work possible. I also want to thank all of you for being here, for listening. Here's my email if you'd like to contact me, and I would love to answer your questions at the appropriate time. Thank you.